We're very excited today to have Bilal Haider talking to us. Bilal, one of the newest faculty members in BME, did his undergraduate work at the University of Illinois, then headed east to Yale to do his PhD work with David McCormick, and then kept going east across the pond to University College London to do a postdoc. That was joint with Michael Heuser and Matteo Carandini, right? So a couple of superstars, obviously, who I'm sure he was able to impart a lot of wisdom to while absorbing a few things before coming back west to join us here. So Bilal joined the faculty in January, is that right? Mm-hmm. And has been frantically building a lab and getting all set to go on the ground here. So we're excited to hear what he has to tell us about here today.
All right. Thanks, Chris, and thanks for putting the seminar series together. It's been really good so far, and I think it's going to get even better as we go into the spring. So, can everyone hear me through the microphone? Okay. So the title of my talk today is Inhibitory Control of Cortical Activity in Vivo. Last week we heard from Barry Connors mainly about the thalamus and its relationship to the cortex. Today I'm going to tell you all about cortex. And I'm just going to start a little high level. Why do we care about the cortex in the first place? So the cortex is this wrinkly outer part of the brain, which is common for most pictures of the brain. It sees its greatest expansion in primates as we move through mammalian evolution, and in particular in humans. And if we just look at the mass of brain structures in the human brain, the cortex is 82% of the human brain by weight. So when we talk about the human brain and human brain dysfunction, it's often dominated by the cortex. But even in smaller species, such as the mouse, which we'll get to towards the end of the talk, even the mouse, almost half of its brain is cortex by weight. So throughout mammalian evolution, the cortex is really a major part of the brain. And interestingly, the mouse is more related to humans evolutionarily than cats, for example. We'll also talk about cats. So despite these large differences in the size of the cortex as we go across species, when we actually look at the elements of the cortex, they're remarkably conserved across species and across cortical areas. And the neurons in the cortex basically fall into two broad categories. The first category are the excitatory cells. They're typically pyramidal in shape, and these cells comprise 80% of the cortex, the total number of cells. They're pyramidal in shape usually. They're extensively branched. They're found all across layers. We heard about some of the layers last week from Barry Connors. And they're massively interconnected with each other, both within layers and across layers. They're also the cells that send information out of the cortex to cause movement, for example. So these are the cells that project to spinal cord, for example, but they project to thalamus as well. The other major type of neurons in the cortex are the inhibitory neurons. These neurons comprise only 20% of the cortex, but as we'll see, they're actually very important for cortical function. They typically don't send projections long range out of their local area. They're heavily interconnected with one another and with other pyramidal cells, but they keep their axons within the cortex. This is why they're called interneurons usually. Okay? So there's great diversity morphologically for both of these cell types. What we're interested in is in function. How are functions in these cell types related, and how can we get a handle on the basic operations in circuits of these kinds of neurons? So what we're going to do is we're going to simplify things and focus on two major types of these excitatory and inhibitory classes of neurons. The first type that we'll focus on are the so-called fast spiking interneurons. These interneurons comprise 30 to 40% of all the interneuron population, so they're really a major component of the inhibitory system in the cortex. And the excitatory cells that we'll focus on are the regular spiking pyramidal cells, and these pyramidal cells comprise 60% of all of the excitatory cells. Okay, so these two circuit elements really form important building blocks for all cortical circuits across areas and across species. So what we're really interested in, as I said, is function. We're interested in how this microcircuit functions as we look at different patterns of activity in the cortex. So what I'm going to tell you about today is how this circuit responds and how this circuit changes as a function of differing levels of arousal. So the first thing I'm going to tell you about are properties of this circuit during spontaneous activity in the absence of anything happening, any sensory input. And then we'll look at how the circuit responds to natural sensory stimulation, visual stimulation. And these two studies that I'll tell you about are performed in the anesthetized cortex. We'll then take these examples and test them in relevant conditions of wakefulness. And we'll see how the cortex responds and how the circuit responds to sensory stimulation in awake conditions. Okay? And I forgot to say, if there are any questions, please stop me and ask. All right, so the first thing we'll look at is spontaneous activity. And we would like to examine a form of spontaneous activity that's present in the cortex naturally. And one of the most well-studied examples of spontaneous activity is the cortical slow oscillation, which is expressed normally, naturally during slow wave sleep, and also during certain forms of anesthesia. And the cortical slow oscillation is named thusly because there's alternation between patterns of activity and inactivity about 1 second or so in duration, making this a slow oscillation. This pattern of activity is highly synchronized across the cortex during sleep and during anesthesia, and it's easily measured through measures of population activity, such as the local field potential, which is similar to EEG recording, which reports the activity of large populations of mostly pyramidal cells. So this is a pretty easy signal to measure at the level of populations. But we're interested in how single cells represent this activity during the slow oscillation, and in particular, what are the synaptic inputs into the excitatory cells, the pyramidal cells, during this form of spontaneous activity. Okay, so you can imagine that this kind of slow oscillation could be set up in one of two ways. One could be there's an alternation of excitation and inhibition. The periods where we can record activity in the cortex are when excitatory cells are active, and the periods of silence when there's inactivity could be when there's a lot of inhibitory tone. So we have an alternation of excitation and inhibition. A second possibility is that there's actually coactive excitation and inhibition, so the two rise and fall together. And we want to know how synaptic inputs, excitatory and inhibitory, that are coming into single cells represent these two kinds of activity, excitatory and inhibitory. Okay, so to answer this question, we performed recordings of large-scale network activity, the local field potential, as I just showed you, and simultaneously performed intracellular recordings from these regular spiking pyramidal neurons. So this is now an electrical recording of the membrane potential of a single cell, where we can record the subthreshold inputs and the action potential outputs. And at the same time, we record the local field potential activity, which reports the large-scale population activity surrounding this cell. And these are recordings in anesthetized ferret prefrontal cortex. And what you can see is that during segments of this slow oscillation, there's alternation between periods of activity called up states and periods of inactivity called down states, where there's this rhythmic alternation between active and inactive periods. And at the same time, when we look at the membrane potential of a single cell, we see that they're basically in phase with the local network. So when the local network is active, single neuron membrane potential depolarizes, there's action potentials, and when the local network is inactive, there's hyperpolarization and no firing. So if we look closely at the single neuron membrane potential in these regular spiking pyramidal neurons, we see that they depolarize by 15 to 20 millivolts, but then during this portion of cortical activity, these up states, the membrane potential is very stable and stays at this kind of tonically depolarized state before falling back down in the down state. Okay, so as I posited before, these periods of inactivity may be caused by activation of inhibitory circuits that quiet the network. So to answer this question, we directly recorded from fast spiking interneurons during the slow oscillation, and what you can see is that this major class of interneuron is also active in phase with the local network. So when cortical networks are active during these upstates, the interneurons are also active, firing action potentials. And when local networks are silent, we see that the inhibitory neurons are also hyperpolarized and silent, not firing action potentials. So what this tells us is that during cortical activity, both excitation and inhibition are active at the same time. So what does this mean for the synaptic inputs coming into a single pyramidal cell? How is it balancing these excitatory and inhibitory influences?
As you try to unravel these principles of the circuit design and performance, do you think it matters what region of the brain you're in, or what layer of the cortex, or are these overarching principles?
So layers and areas certainly matter. For this particular kind of activity, people have recorded across layers, across areas simultaneously. And during these kinds of really synchronized activities, there's not big differences. So there's subtle differences, but by and large, you see activation of interneurons and excitatory neurons at the same time. Other questions? Okay, so how can we sort of tease apart the excitatory and inhibitory inputs that are coming into a single pyramidal cell? So the first thing that we want to do is we want to make sure that we're only recording the synaptic activity that's coming in to a single neuron that we're recording from. So the first thing that we can do with intracellular recording is we have control over the chemical environment inside the cell that we're recording from, and we can introduce pharmacological agents that block the intrinsic currents inside of a single cell. And these are mainly sodium and potassium currents that give rise to spiking, for example. So by blocking the cell's intrinsic currents, now the activity that we record is mainly due to the synaptic inputs that are coming in to the cell, because we've eliminated the intrinsic activity of the cell we're recording from. Okay. Now, a second advantage of the intracellular technique is we also have electrical control over the cell that we're recording from. Okay, so now that we have blocked the intrinsic activity of the cell, we can also move the membrane potential, control the membrane potential, so that we can better isolate the excitatory and inhibitory contributions to the activity we're recording from. Okay, so in this example, if we hold the cell near minus 75 millivolts, we can ensure that the driving force for the inhibitory activity is minimal, because the inhibitory synapses want to bring the cell to minus 75 millivolts. So by holding the cell near minus 75, we minimize the driving force for inhibition. And therefore, what we record is mainly due to the excitatory inputs coming into the cell. Okay? And we can do the converse experiment. We can now hold the cell near the reversal potential for excitation. And when we do this, we minimize the driving force for the excitatory synapses. So the activity that we record in this condition is mainly due to the inhibitory inputs, because the driving force for inhibition is very small. Its contribution to the activity we record is minimal, and the activity is therefore dominated by the inhibitory synaptic inputs. Okay, so by using the intracellular recording technique with this combination of isolating the synaptic inputs, we can turn this single neuron into a radio that listens to the network in real time. And we can dial it to these two different channels, once to listen to the excitatory inputs, and then in the other condition to listen to the inhibitory inputs. Okay, so in this way we can listen to what the network is doing and its impact on the single cell. Okay, is that clear? Okay, so when we perform this procedure, what we find is that the transition in the cortex from periods of inactivity to activity rapidly engage excitation first in red, but very quickly thereafter, within 20 or 30 milliseconds, the inhibitory conductance, the inhibitory inputs coming into the pyramidal cells rapidly catch up, and then thereafter, for hundreds of milliseconds, the two stay at a stable level with each other. So the excitatory and inhibitory inputs remain at the same level for hundreds of milliseconds while the cortex is in this active state, this up state. So what we see is that the excitatory and inhibitory inputs are balanced with each other when the cortex is spontaneously active like this. The consequence of this balanced excitation and inhibition is that it keeps the membrane potential at a stable level. It keeps it depolarized, and the distance to action potential threshold is only a few millivolts compared to 15 or 20 millivolts every single time to generate a spike. So this enables the cortex to stay in an excited state where small inputs can cause action potentials, as opposed to having very large inputs need to traverse this 15 or 20 millivolts every single time. Okay? And these were the first in vivo recordings of excitation and inhibition during this kind of activity, and they confirmed a lot of theory that came before these experiments that posited that excitation and inhibition have to be balanced to get this kind of stable depolarization and irregular action potential firing. Okay, so the first part shows us that spontaneous cortical activity is composed of balanced excitation and inhibition. This begs the question now, what happens to this circuit when it's confronted by a sensory input? And it's been long known since the first recordings of sensory responses in the cortex by Hubel and Wiesel that the properties of the sensory stimulus greatly affect the responses that you record in the cortex. So in this example, Hubel and Wiesel showed that the size of a bar that they're presenting on a screen to a cat while they're recording in cat visual cortex greatly changes the response that you can record. Okay, so the square here delimits the receptive field of the neuron, and the receptive field of the neuron is defined as the stimulus, the length of the stimulus that causes the greatest action potential response. Okay, so here the bar is barely filling the receptive field. Here it's a little bit longer, we get more spikes. Here it's just filling the receptive field, there's the most number of spikes. But then something interesting happens when you keep increasing the size of the bar. You increase it a little bit, the action potential response goes down. You increase it more, there's only 2. Finally, when the bar is very large, there's only 1 spike elicited, even though there's a stimulus inside the receptive field. That's the best stimulus for this cell. The number of spikes you get is basically the same as having almost nothing in the receptive field. Okay, so this observation has been widely known. It's variously called size tuning or surround suppression, but the mechanism for how this was happening was elusive for many, many years. And you can imagine that there's 2 possibilities. One is that as you increase the size of the stimulus, you decrease the excitation that is coming into the cell. The other possibility is that as you increase the size of the stimulus, you actually increase the amount of inhibition that's coming into the cell. So we wanted to address this question in cat visual cortex. So we performed recordings now of network activity and intracellular activity in cat primary visual cortex while they were viewing stimuli that we could control on a computer screen. So the first thing we did was, for a particular neuron that we were recording from, we mapped the receptive field, the part of visual space where it had its greatest sensitivity for visual stimuli, just how Hubel and Wiesel did, with flashed bars or flash dots. And we defined the size of the portion of visual space where the neuron had its greatest sensitivity for visual stimuli, and this is called the classical receptive field. So then we performed an experiment where we presented stimuli to the neuron under study in 2 different configurations. In the first, we presented a natural stimulus, a natural movie, varying in time, just to the receptive field of the neuron. And in the second configuration, we showed the same movie to the same neuron, but we increased the size of the stimulation field on the screen. Okay, so the movie was just covered with a mask that was the size of the receptive field in this condition, and in the other condition we just increased the size of the aperture to stimulate more of the visual field. Yes?
Why did you use a natural movie instead of just a bar?
Good question. One, natural stimuli are not bars, so we wanted to look at the responses of the circuit when it's confronted by a spatiotemporally rich stimulus. And 2, we were inspired by previous studies in monkeys that showed that natural stimuli evoke really different dynamics compared to bars. The other thing is if you have just a bar, it has a particular orientation and a particular spatial frequency, so it only stimulates a limited portion of the cortex and thalamus and retina, whereas a natural scene contains a lot more energy at orientations and spatial frequency, so it stimulates more of the cortex and the visual system in a natural way. Okay? So we presented these movies in these two different configurations. The important thing is that the stimuli inside the receptive field are exactly the same in the two conditions. Okay? The only thing that's different is now how much of the surrounding portions of visual space we're stimulating, which are called the non-classical portions of the receptive field, the NCRF. Okay? All right, so now let's take a look at what happens in these two major elements of the cortical circuit, the regular spiking pyramidal cells and the interneurons. So what you'll see in the first movie here is the response across multiple trials to the exact same movie that is stimulating just the receptive field of the neuron. We see that across trials there's a lot of depolarization, there's a lot of spikes, the neuron is clearly driven, but there's a lot of variability trial to trial, and it's kind of hard to pick out where exactly the driving portions of the movie are. So now let's look at what happens when we present the same movie to the same neuron, but now all we've done is increase the size of the aperture so that we stimulate the receptive field plus the surrounding parts of the visual field. So you can see the response is dramatically different, even though the stimulus inside the receptive field is exactly the same. Right? We see that the action potential response is greatly reduced in amplitude. There's fewer spikes, which means the neuron is more selective for particular features of the movie. We also see that there's less variability trial to trial compared to just stimulating the receptive field. So the membrane potential trajectory is highly repeatable across trials of the same movie. And we also see, as indicated by the dashed lines here, that there's an overall hyperpolarization of the cell compared to when you just stimulate the classical receptive field. Okay, so these observations of increased selectivity, which we measured with this sparseness metric, the increase in selectivity with the wide field movie was reliable in all the regular spiking cells we recorded from, and at the same time there was also, on average, a significant hyperpolarization in parallel with this increase in selectivity. Okay, so we hypothesized that perhaps there's an increase in inhibitory activity in these wide field configurations that might be mediating these effects. Yes?
So my question is, do you think any of that response just has something to do with the scale of the motion in the movies? So for example, if you used a large field movie but with the same scale down small motion, what kind of effect do you use?
Yeah, so we presented lots of different movies with different temporal, major temporal features, and the effect didn't seem to depend much on the exact temporal frequency of the motion. So you would see this with slowly varying features, with fast varying features, a mix of the two. It really seemed to depend more on the spatial coverage as opposed to the temporal features in the movie. But it's a good question. Okay, so we use this procedure to separate the excitatory and inhibitory inputs that are coming into these cells during presentation of these movies in the two different configurations. And what this recording shows is the excitatory potentials coming into a cell during presentation of the movie just to the receptive field, shown in black, and in the wide field configuration, shown in red. And what you can see is that on average, the level of the excitatory inputs is not that different. There's slight differences in the variance and other things, but on average, the level of the excitation coming into the cell is the same across the two conditions. So now when we look at the same cell and look at the inhibitory inputs coming into it, what we see is that there's a dramatic difference in the inhibition in the two different configurations. So here, since we're recording in current clamp, the increase in inhibition is downwards. So the downward deflections in blue are larger in the wide field configuration compared to when we just stimulate the classical receptive field. Okay, so there's a much larger change in the inhibition as compared to the excitation. These larger increases in inhibition were reliable across the population that we recorded from, with much smaller changes in excitation. And it's important also to note that the increase in inhibition in this wide field configuration is not just a simple scaling of the classical receptive field response. It's not like you just double the response. It's actually also different at different epochs in time, where some portions of the movie are evoking much more inhibition, whereas other portions of the movie don't evoke much more inhibition. So it's actually dynamic in time, depending on what the features of the movie are. Okay? So we hypothesized that there is a particular subtype of interneuron that is mediating these inhibitory inputs that are coming into these pyramidal cells. Is there a question? So we performed direct recordings from fast spiking interneurons and presented the movies in the same two different configurations. And now what you see is that the responses of the fast spiking interneurons are the opposite of the pyramidal cells. When we increase the size of the stimulus, the amount of spikes that we evoke from inhibitory interneurons is much larger, and the overall action potential response during the movie is greater compared to when it's just stimulating the classical receptive field. It's also important to note that even though there's an increase in the amount of spikes that are evoked, the timing of the spikes is very well preserved. Okay? So if you actually just look at where the spikes are being evoked, they're being evoked in these very discrete windows of time during the stimulation of the movie, which matches the inhibitory potentials that we recorded in the pyramidal cells.
You may have said this, sorry, but does it matter where that inhibitory interneuron was relative to classical receptive field and non-classical? Could it be anywhere in that?
So when we do the recordings, we center the movie for the receptive field of the neuron under study. But I think maybe what you're asking is something about the sizes of the receptive fields. So when we measure the size of the receptive fields, these guys have larger receptive fields than the pyramidal cells. So they integrate over larger areas, but we center it just on their receptive field. Yeah.
So I have a similar question. I guess what I interpreted was that you were just recruiting more of the centers around. You're recruiting more interneurons in the surround. And so that's how I was interpreting increased inhibition.
Yeah, that's part of
you
it.
might have a spatial smear of how various interneurons are behaving, unless they're coupled for some reason.
Yeah. So yeah, I don't have time to go into this, but we made a model of this, and the other people have made models. The larger receptive fields of interneurons, when you have a real stimulus which covers the whole visual field, they have a different role in the circuit as compared to when you just stimulate a limited portion of the visual field, which is what most visual studies have done. This is also why we use this stimulus.
Neurons in different locations to behave differently.
Yeah, absolutely. But it's not that the excitatory neurons are the ones driving this whole thing. It's the interneurons that are doing it. Yeah. Okay, so the second conclusion from the studies that I just showed you are that natural visual stimulation strongly activates cortical inhibition, and in particular, fast spiking interneurons. They seem to really be drivers of the effects of increased selectivity and the increased reliability and temporal precision that we see with wide field stimuli as recorded in pyramidal cells. This also confirms some long-standing theoretical predictions about sparse coding and the role of inhibition in sparse coding with naturalistic stimuli by Horace Barlow. Okay, so these two studies that I've showed you about spontaneous activity and natural sensory stimulation were in the anesthetized cortex, an anesthetized ferret and an anesthetized cat. So it was a big question how these excitatory and inhibitory synaptic events translate to awake cortex and awake sensory stimulation. So to answer this question, we now move to mice, where we developed a preparation to record awake synaptic activity from single neurons, and we can directly compare them to responses recorded in the anesthetized mouse in the exact same preparation, the exact same configuration. So we have a one-to-one comparison of the effects of anesthesia and wakefulness on the responses in cortex. The stimulus we used here was not a natural movie. We used bars. We went back to a simple stimulus for two reasons. One, we wanted to present the stimulus very briefly in time, so localized in time with a long interval afterwards, so that we could unambiguously interpret the responses that come after one particular stimulus. And the second reason was we can parametrically vary in space where the bar appears. So the bar will appear randomly in a particular portion in space, and it covers a large part of the mouse visual field. So the stimulus allows us to follow responses in time and in space very clearly. And again, we'll record these large-scale network events through the LFP and also record intracellular activity from regular spiking pyramidal neurons. Okay, so let's look at what happens when we present one of these briefly flashed bars during anesthesia and record the membrane potential of a pyramidal cell. So what we can see is in gray are individual trials to repeated presentation of the bar in the same spatial location. In this case, it was at the center of the receptive field. And what we can see is that the stimulus comes on and goes off in 100 milliseconds, but the response in cortex is very prolonged. There's these long-lasting depolarizations with lots of evoked spikes, and it takes almost half a second for the membrane potential to come back down to baseline levels. Okay, so the stimulus is gone here, but the depolarization in cortex persists for a very long time after the stimulus. Now let's look at what happens in the awake cortex when we present this exact same stimulus in the exact same cell types, but now the animal's awake. So the response looks dramatically different now, right? The depolarization is very localized in time. It comes and goes in less than 200 milliseconds. And the amount of spikes that are evoked is also greatly reduced compared to the anesthetized response to this very same stimulus. So one of the primary effects of wakefulness on cortical responses is to greatly reduce the temporal response in visual cortex to a brief visual stimulus. This effect of wakefulness on duration was observed across all the recordings, where the average depolarization from baseline and then back to baseline was in less than 200 milliseconds during wakefulness, whereas during anesthesia it was lasting almost half a second. Okay, so the effect of wakefulness here is to reduce the temporal duration of response to a very brief stimulus. Now, remember I told you that the stimulus is not just presented in one part in space, it's also presented across large portions of the visual field. So we also wanted to see what the effect of wakefulness is on the spatial responses to these briefly flashed inputs. So the traces that I just showed you were for responses to a stimulus presented at the center of the receptive field, shown by this dashed line, but we presented the stimuli in many parts of the visual field to the left or to the right of the central best location. And what we can see is during anesthesia, not surprisingly, the stimulus at the center of the receptive field is the one that evokes the largest depolarization. But even if we present stimuli off to the sides of the center of the receptive field, we still get a significant amount of depolarization that is progressively delayed in time as we move away from the center of the receptive field. When we present stimuli across large portions of the visual field during wakefulness, instead what we see is that there's really only one and maybe two locations to the side of the center of the receptive field that evoke a significant amount of depolarization. Presenting stimuli outside of the receptive field center doesn't evoke much depolarization compared to depolarization you get during anesthesia. Okay? And you can see that the responses are also much shorter in time compared to the anesthetized, as I already showed you. So we again hypothesized that inhibitory circuits might be playing a major role here in controlling not only the temporal aspect of the response, but also the spatial aspect of the response during wakefulness. Yeah?
Can the mouse move its eyes when it's awake? Like, won't that affect where the receptive field is?
Yes, it could. And if a mouse, so we monitor the eye movements not here, but in other experiments, they move their eyes mostly when they're moving. Running, these animals were stationary, so they moved their eyes very rarely, maybe once every 20 seconds. If the animal moves its eyes in the awake condition, that's only going to make your quantification worse, right? It's only going to make your receptive field broader if you're moving the center, right? So if eye movements were a role here, it's actually making this look worse than it actually is, right?
Another question about the methods here. So you have 14 recordings throughout these past slides, and I'm just curious, do you have the same cell for both anesthetized and awake?
No. So sorry, I didn't explain that. So the anesthetized experiments are totally separate and non-overlapping with the awake experiments. The awake experiments were in animals that are trained to be comfortable and still during head fixation, and they've not experienced any anesthesia during the course of the recording. So there's papers out there that just turn off the anesthesia and say it's awake, but we went the extra miles and did it without any sort of anesthesia. Okay, so let's take a look at what's happening to the synaptic inputs during anesthesia and during wakefulness in these two different cases of visual responses being restricted in space and time. So when we record the excitation and inhibition coming into these regular spiking cells during anesthesia, when we present a stimulus at the center of the receptive field, we see that there's a rapid increase in excitation in red, an increase in inhibition in blue thereafter, and then the two kind of remain above baseline and balanced with each other for hundreds of milliseconds. This is very similar to the activity that we saw earlier in the talk during the spontaneous activity that I showed you. This increase of excitation and inhibition, and then the two kind of stay together with each other. But here, it's evoked by a visual stimulus. So now let's look what happens to excitation and inhibition when we look at the response to a visual stimulus during wakefulness. So what you can see are two dramatically different things. One, the inhibition is bigger than the excitation for the duration of the response. The second thing you can see is that the inhibition lasts longer than the excitation, and they both go below baseline within 200 milliseconds. So this mirrors what we recorded at the level of the membrane potential, where we saw these very short-lasting responses. And what we see is that the level of inhibition is greater than the level of excitation during wakefulness for stimuli at the center of the receptive field. So now let's look at what happens when we present stimuli outside of the center of the receptive field. Okay, remember during wakefulness we saw that there was very little depolarization evoked by stimuli outside of the center of the receptive field. So what we see during anesthesia is that a stimulus outside of the center evokes this sort of delayed increase in excitation and inhibition that again stays elevated above baseline long after the stimulus is gone. Instead, when we present a stimulus during wakefulness, we see there is a much larger increase in inhibition compared to excitation, and this is dramatically different than what is seen during anesthesia. It's pretty difficult to predict that you would see this during wakefulness if you just saw this trace. So there's a dramatically qualitatively different in the level of the inhibitory activity that you record for stimuli in large portions of visual space during wakefulness as compared to during anesthesia. So these were the first recordings of membrane potential and the synaptic inputs in visual cortex during wakefulness. And what we observed was that the level of inhibition is much larger during wakefulness compared to excitation, whereas during anesthesia, the level of excitation and inhibition are basically on top of each other. They're balanced, both for amplitude and for their stimulus selectivity. During wakefulness, the inhibitory selectivity is much broader for where the stimulus is, and the level of inhibition is much greater. So we think that this increase in the level of inhibition really plays a role in restricting the spatial spread of responses in awake visual cortex, and also in restricting the temporal persistence of responses. So the effect is it really makes vision sharper during wakefulness. It makes receptive fields smaller in space and time, and there's a circuit basis for this, which is not observed during anesthesia. So, yes, question?
In the temporal spatial plot that you showed introducing this section, there are a few things that are really evident. One is the spatial sharpening, one is the shorter duration. But the other thing that I haven't heard you mention is the onset of the signals was much faster in the awake condition. And even in the previous slide where you broke out the different component pieces, I didn't see anything that looked to me like it was going to cause faster onset of activity during wakefulness. Do you have any sense of what's happening there? It seems less likely to be an inhibitory.
For the onsets, so it's not just the cortex that's anesthetized. It's also the thalamus, and it's also, the signals from the retina, right? So the effects of anesthesia probably add up from the retinal activity, the thalamic activity, to cortical activity during anesthesia. During wakefulness, all that is probably faster. So the arrival of the signals is probably faster. Okay, so these were the effects of, how much time do we have? Okay. These were the effects of wakefulness on inhibition as viewed from single cells. We also, I don't have time to go into the details here, but we also saw these responses dramatically affecting the network activity, these local field potential responses. So here you can see the local field potential response is also much broader in space and longer lasting in time during anesthesia as compared to during wakefulness. It's much more restricted here. I'm going to have to blow through this section to get to the other stuff. So we recently published a study where we looked at how much information there is about synaptic activity from these population signals from the LFP. And briefly, what we did is we developed a new method to estimate the best linear filter to predict synaptic activity from these local field potential signals. And we can then use these filters to basically quantify how well coupled networks are as measured in the local field potential to the synaptic activity inside single cells. And briefly, what we observed is that during anesthesia, the predictions from network activity for excitation are much larger than they are for inhibition, and also the predictability of sensory signals versus spontaneous signals is not different during anesthesia. Whereas during wakefulness, the predictability of inhibitory inputs is much greater than the excitatory inputs. And we also see that the inhibitory inputs tell you when a stimulus was there versus when there was no activity. So there's also this effect of inhibition that pervades throughout the network. It's not just a single cell thing, it's a whole network thing, which is affecting hundreds, thousands of cells. Okay, so the conclusion from these sort of three studies that I showed you as we go from spontaneous activity to natural sensory stimulation to awake sensory stimulation is that as you increase the level of activation in the cortex, as you increase the level of arousal, paradoxically what happens is you don't increase the level of excitation. What you do is you increase the level of inhibition. Okay, so the recruitment of inhibition seems to be the main sort of factor here as we move through these increases in arousal in cortical activation. Yes?
Is there a pharmacological basis for this? Like, for example, does the particular type of anesthesia that you use specifically target inhibitory circuits?
So we used 3 different types of anesthesia. The effects were the same, using different pharmacological routes of anesthesia. One thing that could be playing a role here, probably is playing a role, is you don't just anesthetize the cortex, you anesthetize everything. So you're greatly affecting the neuromodulatory systems during anesthesia compared to wakefulness. So a lot of the inhibitory system is directly responsive to neuromodulation. So neuromodulation could be a big role here. Okay, so this during this study that I just showed you, the animals were awake, but they weren't doing anything. The sensory stimuli didn't mean anything to the animal. It didn't have to process them in any way. So the thing that we're going to do in my lab now is push along this axis of increasing cortical activation and ask how excitatory and inhibitory circuits are engaged during behavior, during perceptual behavior, when the sensory stimuli actually mean something to the animal and when it informs its behavior. So for this, we've developed a very simple visual task where the animals have to tell us when they see something by licking for a reward. So a target appears on the screen, it appears unpredictably, and the animal learns over the course of training that this target predicts the availability of water. So he learns to lick in response to the target, and that's our way of asking the mouse, what did you see? He tells us by licking. The animals learn this in a couple of weeks, and this just shows a histogram of the average licking in response to the onset of a stimulus. You can see that there's a fairly short reaction time across 50 trials here, where the animal starts increasing licking and gets a reward soon after the first lick. This task allows us to move the location of the target across large portions of visual space. So in this plot, the target was in front of the animal. We can also put the target to the side of the animal, and he also detects it a little bit slower, but he detects it with increases in licking. So this gives us a very simple behavior where we can now look at how spatial expectation and reward in different parts of visual space engages the excitatory and inhibitory circuits, and in particular how inhibition might be controlling sensitivity to different parts of space during behavior. So this is much cooler if I show you a movie of the animals doing this, which I think should work. There we go. So the target should appear here. There it is. And then you see the mouse lick when he detects the stimulus. And then in the second part, you can hear the neural activity while he's doing this task. So there's an increase in firing when the target appears. What we're really interested in now is understanding what happens to this circuit when the animal detects a stimulus versus when he doesn't detect a stimulus. Okay, so how does the level of inhibition and excitation control this? So there's a stimulus that he detected. There's a clear increase in the response in visual cortex. There's a second one. There's a third one. So now watch what happens on this fourth one. There's a response in visual cortex, but he doesn't lick. And then he licks on the very next one. So the activity changes sort of dramatically trial by trial. And we want to know how populations of excitatory and inhibitory cells are mediating these effects, and not just in visual cortex, but further down the chain of behavior, how this is happening in motor cortex as well. Okay, so this task allows us to do these very large-scale recordings of population activity. These are these are action potentials from individual neurons recorded across the depth of cortex. Each row here is a single neuron, and we can record these during behavior, as you just heard. This is an example of 45 neurons recorded simultaneously, where one portion of the recording electrode is sensitive to stimuli on the peripheral part of space, and the other portion of the electrode is sensitive to stimuli in front of the animal. So this gives us a way to look at networks as they're functionally changing during behavior. This task also allows us to record the synaptic inputs coming into single cells. So here we have an example of recordings from a regular spiking pyramidal neuron during perceptual behavior. You can see that the membrane potential is dramatically different on trials where the animal misses the stimulus. There are these large fluctuations compared to when he detects the stimulus with short reaction times. So this is where we're going with this. We want to understand how excitation and inhibition are controlling these things during behavior as it's fluctuating and as perception is correct versus impaired. So that's our goal, is to try and link these cellular mechanisms in defined circuits like fast spiking interneurons and pyramidal cells, regular spiking that I talked to you about today, and link these to perceptual behaviors and try and gain some insight about the mechanisms that are controlling perceptual behaviors. So I just want to thank my advisors who taught me everything and allowed me to do all this work, particularly Dave McCormick and Jamie Mazur, who was involved in the cat visual cortex study at Yale, and Matteo and Michael at UCL, and my funding. Thank you.
So that was really great. So Emory Brown gave a talk here about a year ago where he was very careful to distinguish between anesthesia versus sleep. Could you comment on that? Because we were sort of talking about wakefulness versus anesthesia. There's a complicated thing in the middle of that.
Yeah, so I mean, anesthesia is sort of easy to define. Sleep is hard to define. And typically monitoring sleep and quantifying sleep requires multiple measures. So especially in rodents, you can't just rely on one measure like the field potential. You need multiple recordings across different brain areas, recordings of the animal's behavior, its eyes, things like this. EMG, some people record EMG. So it's actually a pretty fascinating question to me if the observations in anesthesia are recapitulated during sleep, right? Is it sort of the same thing? Are we unresponsive during sleep in the same ways that the cortex is unresponsive during anesthesia? But training animals to sleep and reliably measuring sleep is kind of a difficult thing. But,
I got one question from Emory.
Yes,
Have a chance.
Emory.
These are fantastic experiments. Without actually getting to see the animal positioning and preparation too well, I would presume that there is a high degree of head or postural control in determining the receptive field. So to what extent do you think such imposed control impacts the mechanisms that you're exploring?
Yeah.
Because I would imagine normally the animal would not necessarily control its head in a fixed posture looking at a receptive field stimulus.
Yeah, you're right. It's unnatural, but it's convenient and basically necessary for us to do these experiments. We have to immobilize the head and fix the head. There is interesting work using two-photon imaging from Jason Kerr's lab where they looked at how rats, not mice, but how rats move their eyes when they're sort of freely navigating. And it seems that they orient themselves to center the binocular portions of the visual field in a particular place. So if you track their head movements and eye movements, you see that they try to keep the binocular portion of the receptive field stable on a particular portion of the environment. So the things that I showed you today were mainly in the monocular port. We only stimulated one eye. But I think looking at the binocular portions of the visual cortex is probably going to be more impacted by this, where you have precedent for the animal trying to adjust its own posture to keep those portions of the visual field aligned with space.
Thank you.
So what would you predict would happen in your awake scenario when you use natural stimuli?
Yeah, great question. We're going to do it. So I think what you would see is a sharpening of this inhibitory effect, and something that depends on the global and sort of local characteristics of motion, of visual motion. So the stimulus wasn't moving here, but for mice, especially, visual motion is a very potent stimulus compared to static stimuli. So I think the motion aspect of the stimulus would recruit the circuit in a different way, and it would depend on sort of the frequencies of motion that are relevant to the animal's behavior, right? So There's been papers recently that showed that sort of static looming stimuli evoke very different visual responses compared to things drifting in the visual field. So this is something that I think would be super interesting to examine. Yeah. All right, thank you.